Hello, this is Professor Torsen. This is the recorded lecture for Chapter 13. On slide 2 is an overview of the experiment performed by Beadle and Tatum that looked at gene expression in Neurospora bacteria. On slide 3 is an overview of gene expression. Gene expression begins with DNA. DNA, we know, is the blueprint for the organism. It is the set of instructions, and the information is encoded in the sequence of nucleotides. The first step in gene expression is transcription. An RNA transcript is transcribed from DNA. The second step in gene expression is translation. The RNA transcript is translated into a polypeptide. We are going to go over these two steps, transcription and translation, in the following slides. Before we continue, let's go over a quick review of DNA. So DNA, we know, is a polymer of nucleotides, and those nucleotides in DNA are adenine, guanine, cytosine, and thymine. So we've seen this slide several times already this semester, in which these red ribbons are highlighting the sugar phosphate backbone of the DNA strands. And we notice that DNA is composed of these two strands that are running in opposite directions. So they are anti-parallel. And I see the 5' end of this left strand is lined up with the 3' end of this right strand. Also notice the hydrogen bonding between those complementary nitrogenous bases. So we see adenine and thymine paired together, and guanine and cytosine paired together. RNA is also a nucleic acid, which is a polymer of nucleotides. It is a little bit different from DNA, as you can see in this figure on slide 5. RNA is similar to DNA in that those nucleotides are composed of a phosphate group, a 5-carbon sugar, and a nitrogenous base. But as you can see in this figure, RNA consists of one strand rather than the two strands in DNA. There is still a 5' and 3' end, of course, and we see that the nucleotides are abbreviated with C, G, A, and U, standing for cytosine, guanine, adenine, and uracil. So C, G, and A are similar as DNA, but if you recall, the 4th nucleotide in DNA is thymine, but in RNA we have uracil. There are actually 3 kinds of RNA: messenger RNA, transfer RNA, and ribosomal RNA. Our focus for this chapter will be on messenger RNA and transfer RNA. Slide 7 shows you a comparison of DNA and RNA. DNA and RNA are very similar to one another. They are both nucleic acids, which are polymers of nucleotides. And again, nucleotides have the phosphate group, the 5-carbon sugar, and the nitrogenous base. And you can see that is the same in DNA and RNA. The notable difference between the two, of course, is that DNA is double-stranded while RNA is a single strand, and the RNA has the nucleotide uracil instead of thymine. So the first step of gene expression is transcription. A molecule of RNA is transcribed from DNA. This process is similar to the way DNA is copied during DNA replication. Here in this figure on slide 8, we see the DNA represented in red and blue. And here we see the separation of those two strands of DNA. Once these two strands of DNA are separated from one another, the RNA can be transcribed. Again, this process of RNA transcription is similar to the process of DNA replication in that we need these two strands of DNA to be temporarily separated in order to access these nucleotides on DNA and use it as a template to generate RNA. RNA is generated from DNA using the complementary base pairing rules. RNA transcription is different from DNA replication in that only one strand of DNA is being transcribed. So one strand of DNA is a template strand, here in red, and the other strand of DNA is the non-template strand. A second important difference between RNA transcription and DNA replication is that no primer is needed. So the enzyme RNA polymerase is able to attach to the template DNA strand and synthesize this molecule of RNA. The second step of gene expression is translation. This is done by the ribosome, which is this large green blob here on slide 9. The ribosome will translate the language of nucleic acids into the language of proteins. So it will translate the nucleotides into amino acids. Slide 10 shows an overview of the process of transcription. During transcription, RNA polymerase is the enzyme that is generating RNA by using DNA as a template. So in this figure, we see part of a chromosome has been magnified. So we're looking at just one part of a chromosome here, and this part of the chromosome happens to have genes A, B, and C. Notice that there is a promoter at the beginning of a gene and a terminator at the end of a gene. So the promoter is a sequence of nucleotides that the RNA polymerase recognizes as the beginning of the gene that it is trying to transcribe. And the RNA polymerase then will attach to this area and then begin transcribing this gene until it reaches the terminator. So the promoter signals the beginning of a gene and the terminator is a signal for the end of the gene. Notice that DNA is double-stranded, so when we look at these two strands of DNA, only one of these strands is a template for the RNA molecule. Once transcription has occurred, the RNA molecule must be modified before translation can occur. If we look on slide 11, we see a cell pictured, and we know that DNA is stored inside the nucleus. So here we see DNA inside the nucleus of this particular cell, and transcription is being represented here. And so here in yellow, we see the immature or pre-mRNA. So this RNA molecule here was synthesized by RNA polymerase, And again, it needs to be modified before it can be used for translation. So modifications that happen after transcription has occurred are called post-transcriptional modifications. And there are 3 modifications that occur: addition of a 5' cap, addition of a poly A tail, and splicing. Only once all 3 of these modifications have occurred is the RNA considered mature and able to exit the nucleus. And remember, the RNA must leave the nucleus for translation to occur, because the ribosomes, which are going to be doing the translation, are out here in the cytosol. Let's look at the post-transcriptional modification of capping. Capping is the addition of a special nucleotide, which is a modified guanosine, to the 5' end. So some people do call this the 5' cap. We can see this represented here on slide 12. Up top is a picture of a portion of a chromosome, and we're looking at just one gene on this long chromosome. So here in the middle, we see the initial RNA transcript, or the immature RNA, or some people call it pre-RNA. And again, capping, splicing, and tailing must occur to create the mature RNA molecule. And here we see the addition of the modified guanosine, this green dot here, which again is the 5' cap. This 5' cap is necessary for this RNA molecule to exit the nucleus, and it helps the ribosome identify this RNA molecule and bind to it to begin the translation process. The poly A tail is added to the 3' end of the RNA molecule. We can see this represented here on the left-hand side of slide 13. The poly A tail is a couple hundred adenines that are added to the 3' end. This poly A tail increases the shelf life of the RNA molecule. So it allows the RNA molecule to survive in the cytosol a little bit longer. And here on the right-hand side, we see the 5' cap on the 5' end and the poly A tail on the 3' end. The final post-transcriptional modification is splicing. During splicing, introns are removed and exons are spliced together. Introns interrupt the genetic instructions, while exons are what we want to keep. Exons are expressed. They are the coding portions of the gene. So here on slide 14, on the left-hand side, we see an image of a particular gene with exons 1, 2, 3, and 4. Green areas are introns, which are not labeled. So during splicing, these green introns will be removed and the exons will be spliced together. Now, these exons can be spliced together in a number of different ways. For example, we see exons 1, 2, and 3 were spliced together, and that might encode a particular protein, for example, protein A. But another possibility is for the exons to be spliced together such that it is exon 1, 2, and 4. And this RNA would have a set of instructions for a slightly different protein here labeled protein B. So alternative splicing enables one gene to encode for multiple proteins. The second step in gene expression is translation. Translation is done by the ribosome. Back in Unit 1, we learned that the ribosome is responsible for protein synthesis, and now we are looking at the details of this process. So here in yellow is the RNA molecule, and here in this orange color is the ribosome. The ribosome has 3 locations or sites: the A site, the P site, and the E site. Notice here in the P site, a purple molecule, which is representing the transfer RNA or tRNA. And that tRNA has the current polypeptide strand that is being generated. So the P site is where the growing polypeptide strand is located. The A site is where the next amino acid will enter the ribosome, and it will enter the ribosome by being carried by its tRNA. So the tRNA with the next amino acid will sit in this A site of the ribosome. And this E site on the left-hand side is where an empty tRNA will leave the ribosome. Slide 16 is showing you this process in a little bit more detail. So here in yellow is the transfer RNA that is holding the growing polypeptide strand. Here we see this transfer RNA with the next amino acid, and this growing polypeptide strand will be transferred from this tRNA to the tRNA on the left-hand side. Translation occurs in 3 basic steps: initiation, elongation, and termination. Initiation begins at the 3' end of the RNA molecule. And remember, there is a specific start codon that encodes for the start or the initial amino acid in the polypeptide. And then elongation is when the next amino acids are added. And finally, termination is when the stop codon is reached. And that signifies the end of the polypeptide and the end of translation. Once translation ends, the polypeptide must be folded correctly by chaperone proteins in order to become a functional protein. So here on slide 17, we see this process of making proteins represented in a figure from your textbook. So in this middle image here, we see the tRNA in the P site has the growing polypeptide strand. Here in the A site, the tRNA has brought in the next amino acid for the polypeptide. This polypeptide strand will be transferred or moved from the tRNA on the left to the tRNA on the right. And at the same time, this ribosome will shift to the right, so that this tRNA that was in the P site will be in the E site and exit or leave the ribosome. This tRNA that was in the A site would then be in the P site and holding the polypeptide strand, and the A site would be empty and ready for the next tRNA to bring in the next amino acid. Within this system, there is redundancy. Because there are 4 different nucleotides and 3 nucleotides in a row is a codon, there are actually 64 different possible codons, which is necessary for 20 different amino acids, as well as the start and stop codons. So if you notice on this figure on slide 18, there is redundancy in the system. And by that, I mean there is more than one codon to signal a specific amino acid. And looking at leucine here as an example, there are 6 different codons that all encode for the amino acid leucine. This redundancy is helpful because minor changes to the nucleotide sequence, minor mutations, may not result in any detrimental effects. So for example, if I was supposed to have the sequence CUU, but there is a mutation that changes my sequence to CUC, it won't be too big a deal because I will still end up with the correct amino acid, and therefore I will still end up with the correct polypeptide. Mutations happen all the time as a result of errors with enzymes like DNA polymerase, as well as exposure to things in the environment like various mutagens or carcinogens. Mutations are any change in the sequence of nucleotides. And if we look at base pair substitutions first, we are looking at a change in a pair of nucleotides. And as I mentioned, one possibility is a silent mutation, in which the mutation or the base pair substitution has no effect. And that is illustrated here on the left-hand side of slide 19. We see that the sequence of nucleotides was supposed to be GAA to encode the amino acid glutamic acid, but there has been a mutation to GAG. However, because of that redundancy in the codon system, we end up with the same amino acid. So there is no change in the sequence of amino acids, and therefore we will still end up with the same functional protein. However, 2 different substitutions will result in a more significant effect. The missense mutation is a base pair substitution that changes one amino acid. So this is shown on the right-hand side of slide 19. Here we're looking at the RNA molecule and the sequence of nucleotides there. And beneath that, we see the sequence of amino acids. Here on the bottom, we see a mutation that has changed this particular nucleotide so that we get a slightly different codon here. Instead of CCU, which is the codon for proline, we end up with ACU, which is the codon for threonine. So with this missense mutation, we end up with one amino acid that is different. This different amino acid will affect the final protein and may result in a non-functional or protein that doesn't function as well. The last possibility is a nonsense mutation. In a nonsense mutation, the base pair substitution results in a codon that was specifying a particular amino acid into a codon that indicates stop. So in this figure here, if this mutation, instead of changing from proline, changed to a stop codon, we would get a shortened or truncated protein that would likely not have its correct function in the cell. Here on slide 20, we are looking at another possible mutation that could occur, which is called frameshift mutation. In frameshift mutations, 1 or 2 pairs of nucleotides are either added or deleted, and this results in a change in the reading frame. And by reading frame, I mean the sequence of 3 nucleotides, or the sequence of nucleotides that indicate a particular codon. This is represented on the figure on the left. We see that the normal nucleotide sequence has a particular reading frame that groups A, C, G together, indicating the amino acid threonine, and then A, G, G together, which is the codon for arginine, etc. But a frameshift mutation shifts the reading frame such that the nucleotides are grouped together differently. So rather than ACG grouped together and then AGG grouped together, we see that the grouping is CGA and then GAA, which results in 2 completely different amino acids from what should be created. So this frameshift mutation results in a more significant change to the resulting polypeptide and will likely result in a non-functional polypeptide. My analogy for this is if you were given the sentence, "the fat cat sat," but you deleted 1 letter, if you still are trying to read the sentence by grouping these letters in 3s to represent the codons, by deleting that 1 letter, you have changed the sequence, or sorry, you have changed the sentence, and it is now no longer readable. This concludes the recorded lecture for Chapter 13. Thanks for watching, and take care.